This video is on Chapter 19, Blood. And so for Chapter 19, 20, and 21, we're talking about the cardiovascular system. Blood is the fluid component of the cardiovascular system. Blood is a specialized connective tissue that has 5 major functions. First, it transports things like dissolved gases, nutrients, hormones, and metabolic wastes. For example, oxygen is transported in the blood from the lungs where you breathed it in to the tissues, and carbon dioxide is transported from the tissues where it's produced to the lungs so you can breathe it out. Nutrients like glucose and amino acids are transported to tissues that need to use it. Hormones are transported from endocrine glands to target cells. And waste products that were produced by tissues are transported to the kidneys so your body can excrete them in urine. The second function of blood is that it regulates the pH and ion composition of the interstitial fluids. So active tissues in your body generate acids, and acids need to be neutralized. So the blood absorbs those acids and then neutralizes them. For example, lactic acid. Your muscles produce lactic acid, and the blood will absorb the lactic acid and then neutralize them. Ions also diffuse between the interstitial fluids and the blood to eliminate any ionic imbalances. The third function of blood is to restrict fluid losses at injury sites. Blood contains enzymes and other factors that promote clotting to prevent the loss of blood. This is called hemostasis, and we'll talk about this at the end of this chapter. The fourth function of blood is to defend the body against pathogens like bacteria and viruses, as well as the toxins that these pathogens might secrete. The blood contains white blood cells and antibodies, and these things will fight infections and foreign organisms. So we'll talk about that during this chapter as well. The fifth function of blood is that it stabilizes body temperature. Your active muscles and tissues will generate heat. The blood absorbs this heat and will distribute it to other tissues. So let's take a look at the composition of blood. So we know that blood is a connective tissue, and we know the different components of the connective tissues. We know that all connective tissues have 3 things. Connective tissues have a ground substance, they have protein fibers, and they have specialized cells. So blood, which is a fluid connective tissue, isn't any different. It also has these 3 components. So let's take a look at these 3 components. So first of all, this is a test tube here, and when you draw blood from a person, you take a needle and you put it into their veins and you draw blood out, and you put that blood into a test tube. First of all, in order to get that blood, we use a venipuncture process where you're taking the needle and placing it into a vein, and then that blood is being drawn into a test tube. Now when you look at the test tube, you're going to see that all of the test tube inside that fluid in there, which is blood, looks red, and it's diffuse and consistent. But we can take that test tube and we can centrifuge it, and centrifuging means we're just spinning it. And as we spin it, we can see that the different components start to separate from each other. The top component that we see will be clear or a little yellowish, and that is called plasma. Plasma makes up 55% of whole blood. So by whole blood, what I mean is all of those components together. Then we'll see below the plasma these components that are called the formed elements. The formed elements are the blood cells, red blood cells and white blood cells, and platelets that are found distributed throughout whole blood. However, when we centrifuge it, the heavier blood cells are going to drop to the bottom. The red blood cells are heavier than the white blood cells. So right below the plasma, we see a tiny little area that we call the buffy coat. And the buffy coat will be a clearish area that contains the white blood cells and the platelets. White blood cells and platelets make up less than 1% of blood. Another name for white blood cells are leukocytes, and another name for platelets are thrombocytes. Beneath the buffy coat then are the red blood cells, and red blood cells make up 45% of the total blood. Another name for the red blood cells are erythrocytes. The white blood cells or leukocytes are involved in body defense, so we'll talk about those this chapter. The platelets or thrombocytes are involved in blood clotting, and we'll talk about that this chapter as well. And the red blood cells or erythrocytes, which are the most abundant blood cells, they transport oxygen and carbon dioxide. So whole blood is made up of all of these components. It's made up of plasma, white blood cells, platelets, and red blood cells. We call the blood cells, the red blood cells and white blood cells, as well as platelets, the formed elements. These formed elements will be produced through a process that's called hemopoiesis. And we'll take a look at hemopoiesis as well. The stem cell, called a hemocytoblast, is a stem cell that can produce any one of these formed elements. The whole blood has a temperature typically of 38 degrees Celsius or 100.4 degrees Fahrenheit. It is 5 times as thick or 5 times as viscous as water. And the pH of blood is slightly alkaline with a pH between 7.35 and 7.45 with an average of 7.4. So what I want to do now is take a look at plasma, and then we'll look at red blood cells, and then we'll look at white blood cells, and we'll finish up the chapter by looking at the process of preventing blood loss, which is called hemostasis. So if I were to draw out a blood vessel here, inside the blood vessel we would have these red blood cells, and we would also have a few white blood cells. And in between the red blood cells and the white blood cells is the liquidy component of blood, which is called plasma. Plasma makes up 46 to 63% of the volume of whole blood. And of this plasma, water makes up 92% of the plasma volume. Surrounding the blood vessel is a fluid called the interstitial fluid. Both the plasma and the interstitial fluid are extracellular fluids because they're not inside those cells, but they are separated by the lining of the blood vessel wall. Both the plasma and the interstitial fluid have about equal concentrations of the major plasma ions like sodium, potassium, chloride. The main difference between the interstitial fluid and the plasma is the difference in concentrations of dissolved proteins and respiratory gases. The proteins that float around in the plasma are called plasma proteins. These plasma proteins are pretty large, and because of their size and their shape, they're not able to cross the membrane of the blood vessel, so they stay in the blood. So these plasma proteins are found in the blood vessels, but they're not found in the interstitial fluid. The liver makes or synthesizes and releases over 90% of these plasma proteins. These plasma proteins include proteins like albumins, globulins, and fibrinogens, as well as various proenzymes. Albumins make up 60% of the plasma proteins. Because they are the most abundant plasma proteins, they are the major contributors of the osmotic pressure of plasma. So what does this mean? This means that albumins make up a lot of the solutes inside the blood, and they draw water into the blood vessel. Remember that water wants to move towards the solutes, so water is moving towards the albumins, which are causing this osmotic pressure and kind of pulling the water towards it. The second type of plasma protein are globulins. Globulins make up 35% of the plasma proteins. These globulins can either be antibodies or they can be transport globulins. Antibodies are also called immunoglobulins. They attack foreign proteins and pathogens that might be harmful to your body. Transport globulins will bind small ions, hormones, and other compounds like drugs and transport them throughout your blood. The third major plasma protein is fibrinogen. Fibrinogen makes up roughly 4% of the plasma proteins, and it functions in blood clotting. Under certain conditions, fibrinogen molecules interact and they form large insoluble strands of fibrin, which are fibrous strands that wrap themselves around platelet plugs, forming a blood clot. So remember that plasma is 92% water. And in addition to the water, we have the plasma proteins. And in addition to plasma proteins, there are also other solutes. There are some nutrients that are floating around in the plasma, nutrients that are used to produce ATP, for growth when the cells need them, and for cell maintenance. They include things like fatty acids, cholesterol and glycerides, which are all lipids, and carbohydrates, which is primarily glucose, and amino acids. There are also electrolytes in the plasma. Electrolytes are charged ions such as sodium, potassium, calcium, magnesium, chloride, bicarbonate, and other ions that are found in plasma. The other thing that's found in plasma are organic wastes. These wastes are carried to sites to be broken down or to be excreted. Some examples of these organic wastes include urea, uric acid, creatinine, bilirubin, and ammonium ions. So all of these things make up plasma. Now you might hear the term serum. Serum is the plasma with the fibrinogen taken out of it. So serum is basically the plasma minus the clotting proteins. So now we're done talking about the plasma, and now I want to talk about the formed elements. The formed elements again are made up of the red blood cells, the white blood cells, and the platelets. So we will cover the red blood cells for the remainder of this video, and then we'll pick up and talk about the white blood cells and the platelets in our next video. The red blood cells are the most abundant formed elements, and they make up 99.9% of the formed elements. 1 microliter of whole blood contains 4.5 to 6.3 million red blood cells in the adult male and 4.2 to 5.5 million red blood cells in the adult female. The difference in red blood cells between the male and the female are because of the testosterone that the adult male produces, which increases the process of erythropoiesis, which is the production of red blood cells. Hematocrit is the percent of blood that's occupied by the cellular elements, or the formed elements, like the red blood cells, white blood cells, and platelets. But since we said that the red blood cells make up 99.9% of the formed elements, basically with a hematocrit, what we're looking at is the percent of red blood cells in the blood. The hematocrit in males averages 46%, and the hematocrit in females averages 42%. Inside each red blood cell are globular proteins called hemoglobin molecules. Hemoglobin makes up over 95% of a red blood cell's intracellular proteins. So let's take a look at one hemoglobin molecule to see what it looks like. So hemoglobin is made up of chains of polypeptides. So here's a chain, and here's a chain, and here's a chain. So we have 4 polypeptides that make up a hemoglobin molecule. Each of the 4 polypeptides in the hemoglobin molecule contain a molecule that's called heme. Heme is a pigment. Each heme contains an ion of iron. The iron can bind to an oxygen molecule, and when it does that, it makes the red blood cell appear red. When the iron isn't bound to oxygen, then the red blood cell will appear a darker red or a blue color. Ultimately, one hemoglobin can carry 4 oxygen. Each red blood cell has about 280 million hemoglobin molecules. So if we multiply 280 million hemoglobin by 4 oxygen, that means that every red blood cell can carry over 1 billion molecules of oxygen. Hemoglobin content of whole blood is reported in grams of hemoglobin per 100 mils of whole blood, or grams per deciliter. The normal range in males is 14 to 18 grams per deciliter, and in females is 12 to 16 grams per deciliter. Okay, so let's talk about the lifespan of the red blood cell. So about 1% of the red blood cells are replaced every day. This means that about 3 million red blood cells have to be produced and then enter into circulation every second. The red blood cell will circulate in the blood vessels for approximately 120 days or 4 months before they are broken down. New red blood cells are formed in red bone marrow in tissue that's called myeloid tissue. This red bone marrow is found in bones like irregular bones like the vertebrae, or flat bones like the sternum, the ribs, the skull, the scapula, the pelvis, and they're also found in the proximal ends of the limb bones. The process of making red blood cells is called erythropoiesis. So we're going to go through some of the stages of red blood cell maturation, and we'll begin at the stem cell. So there's a stem cell that's called a hemocytoblast. And the hemocytoblast is going to divide in the bone marrow, in that red bone marrow, to produce 2 other stem cells. So the hemocytoblast is kind of this pluripotent or omnipotent stem cell that can actually become other stem cells. So the hemocytoblast becomes a myeloid stem cell or a lymphoid stem cell. The myeloid stem cells will divide and produce or differentiate into red blood cells and white blood cells, whereas the lymphoid stem cells will divide and produce the various classes of lymphocytes. Let's look specifically at the red blood cells. The erythroblasts are very immature red blood cells that are actively synthesizing that protein that we call hemoglobin. These erythroblasts have nuclei. After about 4 days, the erythroblasts shed their nuclei and they become a reticulocyte. A reticulocyte is an immature red blood cell. These reticulocytes will enter into the blood vessels, and that's where they will mature into fully mature red blood cells. The reticulocytes account for about 0.8% of circulating erythrocytes. A hormone called erythropoietin, which is abbreviated as EPO, stimulates red blood cell production. So it increases red blood cell production. It also speeds up the maturation of red blood cells. So it's speeding up how fast the reticulocytes become mature red blood cells. erythropoietin is released by the kidneys where it's made whenever the oxygen levels decrease. So the kidneys are monitoring the blood that comes into them. And if the blood flow to the kidneys declines, the kidneys think that there's not as much oxygen in the blood, and so then the kidneys will release the erythropoietin. Other conditions that decrease the oxygen levels coming into the kidneys include the condition called anemia, or when a person is in high altitude and their oxygen content in their lungs declines, then the oxygen content in the blood declines, going to the kidneys can detect that. Or if the respiratory surfaces of the lungs are damaged and less oxygen is coming into the blood. So the kidney is sort of detecting that there's low oxygen for whatever reason. Either there's not enough oxygen in the blood or there's not enough blood coming into the kidneys, and this will trigger the kidneys to release erythropoietin. Erythropoietin then travels in the blood to the red bone marrow, where it increases red blood cell production and speeds up the maturing of the red blood cells. Producing red blood cells requires amino acids, iron, and vitamins B12, B6, and folic acid. The amino acids are necessary to create the proteins or the polypeptides. The iron, we know, is at the center of the heme group. Pernicious anemia is a type of anemia where there is low red blood cell production due to a vitamin B12 deficiency. Iron deficiency anemia is the most common type of anemia and is caused by a shortage of iron in your body. Another type of anemia is sickle cell anemia, where the shape of the red blood cell is distorted, which reduces its oxygen carrying capacity. Red blood cells are small, specialized discs that are thin in the middle and thicker at the edge. The shape of these red blood cells give three important effects. First of all, there's a high surface to volume ratio, meaning there's a lot of surface area for these red blood cells. So this high surface area allows for oxygen to be quickly absorbed and then released. It also allows the red blood cells to form stacks called rouleaux. And it's necessary for these red blood cells to form stacks and to be able to get through really narrow blood vessels called capillaries, where they go through these blood vessels in single file. This shape also allows the red blood cells to bend and flex when they're entering into these small capillaries. In this picture here, using a scanning electron microscope, we can actually see the three-dimensional shape of the red blood cells. However, when we're looking at a smear of blood under a light microscope, then the red blood cells are going to appear as two-dimensional objects because they're flattened against the surface of the slide. And where the middle of the red blood cells are thinner, they will actually appear more as white centers or look like there's holes in those red blood cells. So now let's take a look at blood types. And first we're going to talk about antigens. Surface antigens are proteins that are on the surface of all the cells in your body. And these are substances, these antigens are substances that your immune system recognizes as normal. So your immune system won't attack your cells because they see these antigens as being normal. Blood type is determined by the presence or absence of specific surface antigens on the red blood cell. These specific antigens are called agglutinogens. So these agglutinogens are proteins on the surface of red blood cells that have specific shapes. The agglutinogens that are of particular importance when typing blood are the A, the B, and the RH or D agglutinogens. A person who is considered type A has the A antigen on their red blood cells. A person who is type B has the B antigen on their red blood cells. A person that is type AB has both antigen A and antigen B on the surface of their red blood cells. And a person who is type O has neither antigen A nor antigen B on the surface of their red blood cells. If a person who is type A received blood that is type B, the body would recognize the B antigen as abnormal, and the person with type A would have antibodies in their plasma that would attack the antigen B. The antibodies in the person with type A are called anti-B antibodies, and these antibodies are found in their plasma. The anti-B antibodies will attack the antigen B of the foreign red blood cell. So type A has the antigen A on the surface of their red blood cells, and they have the anti-B antibodies in their plasma. Similarly, a person with type B blood has the antigen B on the surface of their red blood cells, and in their plasma, they have anti-A antibodies. Because if a person that has type B blood came in contact with the A antigen, their anti-A antibodies would attack the A antigen. A person with type AB blood has both antigen A and antigen B on the surface of their red blood cells. But in their plasma, they don't have either antibodies. Because if they had either the anti-A or the anti-B antibodies, their antibodies in their plasma would be attacking their own red blood cell. Type O blood is blood that has neither the A antigen or the B antigen on the surface of their red blood cells. But in their plasma, they have both types of antibodies. They have the anti-A antibodies and the anti-B antibodies. Each person is born with one of these types of blood, and their plasma contains the corresponding antibodies or lack of antibodies from birth. So let's look at the Rh antigen. The Rh antigen is either there or it's not. Another name for the Rh antigen is the D antigen. So if a person has the Rh antigen or the D antigen on the surface of their red blood cells, they would be considered Rh positive. If a person does not have the Rh antigen on the surface of their red blood cells, they are considered Rh negative. A person who is type A can be either Rh positive or Rh negative. Similarly, type B blood can be Rh positive or negative, and so can type AB and type O. When the complete blood type is recorded, the term Rh is usually omitted, so we'll say A positive or A negative, B positive or B negative, and so on. A person who is Rh negative and does not have the Rh antigen on the surface of their red blood cells at some point in their life may develop the Rh antibody. These antibodies, though, are developed through exposure to the Rh antigen. A person that is Rh positive will never develop the anti-Rh antibodies in their plasma, because if they did, then the antibodies in their own blood would be attacking their own red blood cells. If a type A person with the A antigen on their red blood cell and anti-B antibodies in their plasma came in contact with a type B blood where the antigen B was present on the red blood cells, the anti-B antibodies would attack the red blood cells that contained the antigen B, because to these anti-B antibodies, antigen B looks like a foreign red blood cell. So these Y-shaped antibodies will bind to the foreign red blood cells and cause those red blood cells to clump together. The clumping is called agglutination. And so another name that we give to the antibodies in plasma corresponding to blood typing are called agglutinins because they will cause agglutination. When agglutination occurs, it occurs because of a cross-reaction. A cross-reaction is when an antibody meets its specific antigen. So, in other words, when the anti-B antibody meets a red blood cell with the antigen B, or when an anti-A antibody meets a red blood cell with the antigen A. Cross-reactions can occur during blood transfusions. If a person is given an incompatible blood type, a transfusion reaction can occur. During a transfusion reaction, red blood cells will clump and form drifting masses that can plug small blood vessels. These small blood vessels could be in the kidneys, the lungs, the heart, or the brain, and cause major complications. An acute transfusion reaction will typically present with adverse signs and symptoms during or within 24 hours of a blood transfusion. The most frequent reactions are fever, chills, and a rash with round red welts on the skin that itch pretty intensely. These symptoms typically resolved pretty promptly without any specific treatment or complications. However, transfusion reactions that cause these blood clots in the kidneys, lungs, heart, and brain are more serious. So whenever blood transfusions are going to take place, then testing will be performed on both the donor and the recipient blood to test for compatibility. The donor is donating their red blood cells and not their plasma. So it's important to look at the antigens on the donor's red blood cells. The recipient can't have the corresponding antibody in their plasma. So a person that has type B cannot be given to a person that has the anti-B antibodies. So type A blood can be donated to a type A recipient. Type B blood can be donated to a type B recipient. And type AB blood can be donated to a type AB recipient. And type O blood is donated to a type O recipient. However, if there isn't a compatible cross match, then O negative can serve as a universal donor. Remember that type O negative contains no surface antigens at all, so will not cause a reaction in any blood type. In other words, type O doesn't have the A antigen, the B antigen, or the Rh antigen on the surface of the red blood cells. O negative is not a common blood type, however. Before we end this video, I want to talk briefly about the hemolytic disease of the newborn. This disorder occurs when an Rh negative mom is pregnant with an Rh positive baby. So this means that the mom's blood does not have the Rh antigen on the surface of their red blood cells, but the baby that the mom is carrying does have the Rh antigen on his red blood cells. So during the first pregnancy, there's no mixing of blood between mom and baby. The placenta makes sure that there is a barrier between the blood, between mom and baby. And remember, mom is Rh negative. She doesn't carry any of the Rh antigens on her red blood cells, but she also doesn't have any antibodies in her blood, in her plasma either, because she wasn't born with those antibodies. So mom is Rh negative, and the blood's not carrying the anti-Rh antibodies if she hadn't already been exposed to the Rh antigen. Then the baby is born, and during the birth process, the mom's blood and the baby's blood mix together, so the Rh negative blood of mom mixes with the Rh positive blood of baby. This sensitizes the mom's blood, and the mom then starts developing these antibodies against the Rh antigens. Then during a second pregnancy, the antibodies that mom has started to develop can cross the placenta and attack the baby's Rh positive red blood cells. So whereas the red blood cells are too big to cross the placenta, the antibodies are small enough where they can cross the placenta. So the mom's antibodies cross the placenta and they start attacking and causing agglutination of the baby's red blood cells and forming those little clots that will be carried to the lungs and the brain and the heart and cause fatal complications. This ends our first video on Chapter 19, the blood.